They all participated in the UT Southwestern program called STARS this year, or last summer. And basically, it was just researchers with PhDs for like from 40 hours a week to 60 hours a week for 10, I think 10 weeks during the summer. So they worked really hard on this, so try to give them your full attention. So first up is Mason.
Hi guys. Can you hear me well? Yes. Okay, great. So like Andrew said, my name is Mason Amelotte. If you don't already know me, I'm a senior, and I was one of 3 students selected to participate in the UT Southwestern STARS summer research program. So that's what I'm going to be talking about today. First of all, a lot of you probably don't even know what is STARS, so I'm going to kind of explain that. The STARS program is, like I said, UT Southwestern in their biomedical labs. It's an 8-week summer research program under a principal investigator. And a little bit of information about my lab. I was in Dr. Deborah Clegg's lab. She's an associate professor in the Department of General Medicine and Clinical Nutrition, and her lab is in the Touchstone Center for Diabetes Research. And she also studies obesity and estrogen's effect on the 2 diseases. And my mentor was Michael Finus. He is a lab researcher, he's a lab tech in Dr. Clegg's lab. So now we've got kind of a basic understanding of where I spent my summer. I'm going to talk about my project. And the title of my project is Novel Findings Implicate Inverse Relationship Between MT3 and PR alpha. And you guys probably have no idea what that means, but we're going to get there. So right now you're probably asking yourself, what is MT3 and PR alpha? Well, PR alpha stands for estrogen receptor alpha. It's basically a receptor found in cells within the human body, and it's activated by the hormone estrogen. So when estrogen is present in the body, PR alpha is turned on, and it acts as a transcription factor that basically turns on other genes in the body. Yes. Oh. So, what is MT3? Well, MT3 also is a transcription factor in the body, and it stands for metallothionein 3. And I'm going to talk a little bit more about that, but just for the sake of pictorial representation, I made a little cartoon character. Right there, and that's going to basically be MT3. So like I said, what is MT3? It stands for metallothionein 3. It is a gene in the body, and it's found in the cerebrum, it's found in the prostate, the testes, kidney, and the liver. And its function is it controls the uptake regulation of heavy metals in the body. So specifically, this gene binds to metals such as silver, copper, and zinc, and it basically removes them from your body, which is a pretty good job. So I'm going to shift gears and kind of talk about the background of my experiment. So MT3 is a gene, like I said, that is found then lastly, MT3 has been induced by hypoxia in adipose tissue. So what that means is, in fat tissue, when you have really low levels of oxygen, your MT3 levels elevate. So again, trying to find an inverse relationship between MT3 and air output. When you have high levels of MT3, you're chemo-resistant, your cancer cells grow a lot. When you have low levels of air output, the same thing occurs. What's the connection between the two? And this I looked at in fat tissue. So what my lab presented me with was some data on a microarray, which basically does a huge screening of all the genes in a certain tissue sample. And what we found is that in whole adipose tissue, WAT, males had a lot more MT3 than females did. And as you would expect, females have a lot more estrogen than males. So this kind of points to the inverse relationship idea. And so we looked in different components of fat tissue. So we looked in a component called stromal vascular fraction, which is basically just a part of adipose tissue as a whole. And what we did is we had our wild type WT, which was our control sample of the male and female fat tissue. And we also included ERCO samples. ERCO stands for estrogen receptor knockout. So that basically means there's no estrogen present. So it would be this, I guess, manlier tissue. But what we would expect is when you have high levels of MT3, or when you have no estrogen in the body, you would have these high levels of MT3. And that's what we found. So in the stromal vascular fraction of fat, when estrogen receptor alpha is knocked out, you have really high levels of MT3 in both the males and the females. And then as expected, estrogen is higher than the female. That's just kind of used as a control. So next we tested this in adipocytes, and that's your basic fat tissue for fat cells. And again, the same conclusions were drawn. And what we found was, again, MT3 was higher in the ERCOs than in the wild type. And estrogen receptor alpha was higher in the females than the males. And of course, because estrogen receptor alpha is knocked out here, you see no levels of the gene itself. So then we looked at MT3 and estrogen receptor alpha in pre-adipocytes, and those are basically pre-fat cells, or fat cells that haven't yet matured or differentiated into their full adipocyte form. And while we basically had the same experimental setup, we also included a hypoxic group. So you had your male and female, wild type and ERCO pre-adipocytes, but you also had the same group of tissues that were deprived of oxygen for a certain period of time. And in my case, it was 24 hours. And what we did was we basically exposed these pre-fat cells to 1% oxygen, so normal oxygen levels, the air you breathe, 21% oxygen, but we put it in a chamber that only had 1% oxygen. The rest was nitrogen and carbon dioxide. And what we were expecting is can ask about the program too, not just the experiment.
What last year have you like go back to that? Yeah. What did you find from that? I don't understand.
Pardon me?
What did you discover from the protein map?
So what we found is that in these hypoxic